Dr. Dioko Kim. Dr. Kim is an associate professor at Johns Hopkins. His group's research spans the disciplinary boundaries between nanotechnology, biomaterials, and mechanobiology, with an emphasis on their applications to tissue engineering and regenerative medicine. Like Dr. Lorraine, Dr. Kim is also an entrepreneur, having founded Nanosurface Biomedical, which is now Curie Bio. They're focused on integrating human iPSC-derived cells, tissue-specific biosystems, AI-enabled data analytics to accelerate the discovery of new therapeutics. Recently, though, Dr. Kim's team sent their engineered heart tissue platform to the ISS. Dioko, please take it away.
Okay. It is my great pleasure to introduce our tissue chip in space project. My name is Dr. Kim. I am currently associate professor in the Department of Biomedical Engineering here at Johns Hopkins University. So today I will be giving an update on our heart tissue chip in space project and our recent SpaceX 20 launch experiment. Next. As you know, space flight affects almost every physiological system in our human body, including the cardiovascular system. NASA has identified several cardiovascular risks as the area of the high priority for research, including diminished function, occurrence of the serious cardiac arrhythmia, and impaired response to stress and exercise. While we have gained the important insight from the previous study, a shift to using the 3D human in vitro model of the cardiac tissue would significantly help to better understand microgravity effect on the human heart. Further, the findings from this study on microgravity might help inform research on effect of aging here on the Earth. Next. Main goal of our project is to develop the high-throughput microphysiological model of the human myocardium, which is derived from the human-induced proliferative stem cell here in this schematic illustration, so that we can study the effect of the microgravity on the human cardiac tissue structure and function. We have been working on this project over the past 2 years, sponsored by NIH and CASES. And I will be primarily talking about the major progress made during the first phase of our project over the past 2 years, which is to design and validate our micro cardiac tissue model on the Earth, and then send it into the space for 1 month stay on the International Space Station, where their contractile function will be assessed in real time before they retrieved for structural, biochemical, and genomic analysis. Next. So here is our engineered human heart tissue, which is also called as EHT. So we use the EHT platform, which is composed of the iPS-derived cardiomyocyte and the stromal cells that are seated within the hydrogel scaffold, and then cast around the 2 silicon posts, in which deflection of the flexible post here allowed us to measure the contractile force generated by this tissue. You can see the movie of the beating microcardiac tissue within our 24 EHT platform here. Next. Since one of the major roadblocks to using the human iPS-derived cardiomyocyte and tissue for in vitro study is phenotypic immaturization, post-differentiation. We have utilized the novel electroconductive hybrid hydrogel to serve as a scaffold to improve the maturity of the engineered heart tissue. This hydrogel is composed of the decellularized extracellular matrices shown here upper left panel, which is derived from the porcine left ventricular myocardium, and then reduced graphene oxide from the heart tissue with electroconductivity and stiffness of the RGO. We were able to generate the tissue with enhanced functional maturization. Next. As indicated by the increased twitching force and upregulation of the key cardiac gene and improve the electrophysiological function. Next. We further modified our engineered heart tissue platform by incorporating the magnet into the flexible force. This magnet, when coupled with the area of the giant magnet photoresistive or GMR sensor allow for the measurement of the post-deflection in automated and real-time manner. Next, as posts are deflected, the resulting change in the magnetic field is also detected by the sensor, and this can be converted into the voltage signal, then force readout without using the relatively inefficient method of the live tissue imaging by combining the direct sensing here and reference sensing circuit on the board. We are able to subtract out the noise signals to give the clean readout. Next, EHT array and next incorporate into the space flight chamber. They are designed by the BioServe Space Technology, our instrumentation partner. This chamber consists of the hard polymer body, gas permeable PDMS membrane, and self-sealing the pulse for the fluidic exchange. Array of these 6 tissue each can be inserted, and the whole chamber sealed to prevent leakage, with the end result looking something like this in this slide. Next, and this chamber are next integrated into the BioServe plate habitat or PHAB. For this experiment, each PHAB holds 4 chambers, with each chamber pair residing over the printed circuit board containing the magnetic sensor, seen here in this slide. Once chambers are inserted and secured, the entire assembly is sealed in the anodized aluminum case, then contain the band that allow for the gas exchange when the PHAB is situated in the civil incubator that is already in the International Space Station. Next. So this is what integrated PHAB looks like. Next. And here is the Jonathan Sui, my postdoctoral scientist, who is holding the one up for the inspection while at the Kennedy Space Center. And finally, this picture on the right partially demonstrates how PHAB is loaded into the aluminum casing. For this first flight, we used 2 PHAB and 8 chambers in total. Next. So here is our experimental design. Upon arrival at the International Space Station, all tissue are fed, and then subset of the sample are terminated at the day 7, 14, and 28. Termination consists of the fixing in PFA for immunostaining, or preservation for RNA later or RNA sequencing analysis, and subset of tissue will be also returned viable and cultured for the additional week on the Earth to see if there's any recovery effect. Next, with the engineered hard tissue platform and the space flight hardware developed, we also conducted the experiment validating testing, or EVT. So this slide shows our EVT assessment result. So we assessed the entire system and procedure, and this assessment included the long-term tissue viability and validation of the electronics and our software, and chamber integrity during the freezing with the RNA later, and the quality of the resulting immunostaining and the collected RNA. Next. We had initially planned to ship the fabricated tissue to Kennedy Space Station Center in Florida by FedEx overnight shipping. However, our shipping test showed that there was an unacceptably high failure rate with regard to the tissue integrity. So we instead opt to hand transport our tissue to Florida by flight travel. Next. Yeah, so this is our chamber and boxed to be carried over to the Kennedy Space Center at the Florida. Next. So once our engineered hard tissue had arrived at the Kennedy Space Center, they are maintained throughout the pre-launch period, after which final tissue quality check and chamber integration and hardware and software check was conducted prior to handover. On March 5th, early this year, our experiment was handed over to be packed securely away in marine unit, which was a power incubator that would keep our tissue warm and while they made trip up into the space. In this picture, you can see one of our PHAP being strapped in, and entire marine unit would eventually be bolted into the SpaceX Dragon 1 capsule. Finally, shortly before midnight on the March 6th, we was able to see our experiment launched aboard the SpaceX CRS-20 lift supply mission. So this is a real-time movie of the SpaceX 20 launching, which was taken by our science team, which was very exciting moment. Next. So this is a picture of the space astronaut Jessica Meyer during the engineered heart tissue chamber media change and fixing preservation in the life science glove box aboard the International Space Station. And interestingly, Jessica worked with Peter Lee, one of our co-investigator of the science team, in the same lab during her final undergraduate year at the Brown University. What a small world. Next. Yeah, we are excited to find that our engineered heart tissue survived this launch, and they produced the contractile functional data during the 1 month in space. And while the analysis of this data is still preliminary, interestingly, we found that there is already functional deficit due to the microgravity after just 4 days in space, as you can see in this figure. Next. And the twitching force decreased continuously over time, and difference in beat interval variation, which we use as a metrics for the arrhythmia, irregular heartbeat, show a peak at the day 19 in this figure on the right, but the reduction by day 26 and recovery to baseline upon the return to the 1G, which was interesting. Next. So to summarize, we was able to use our novel electroconductive hybrid hydrogel to improve the functional maturity of our engineered heart tissue, and then by modifying our tissue platform to not only enable automated force sensing, but to also be packaged for the space flight. We successfully delivered the experiment to the International Space Station early this year, March, and analysis of the space flight contractor data is still ongoing. We are also currently conducting the ground-based control experiment, as well as the RNA sequencing analysis, which is already delayed due to the COVID-19 associated challenges. We also began to work on the second phase of our project, which is to evaluate whether therapeutic intervention could help ameliorate microgravity-induced cardiomyopathy. So we'll be working on this topic for next 2 years. So we demonstrate that our proposed engineered human heart tissue research model could be potentially serve as an effective tool for better understanding of the cardiac function in response to the external factor such as microgravity, which will be useful for drug development and other applications related to the cardiac dysfunction on the Earth. Next. This is our engineered human heart tissue in space team, which consists of the researchers from the Johns Hopkins University, University of Washington, and Brown University, as well as the BioServe Space Technology, who was our hardware instrumentation partners. Next. Finally, I'd like to thank our funding sponsor, NIH and CASES, as well as the team at the NASA and Space that helped send our payload to the International Space Station. Thank you so much for your attention.